Thank you very much, Mr. Chair, and good morning to all of you. In South Africa, it is currently in the afternoon, so I guess we are a little ahead of you. I think the first thing that I will do in my presentation is just to introduce my co-authors of this presentation. You will recognize the second name on the list there, Dr. Frede Makdama from Espol in Guayaquil. But then we have two co-presenters from Australia, Rhiannon Evans, that is working for Biosecurity Queensland, and Stuart Lindsay, who's working for the Department of Agriculture, Forestry and Fisheries over there. We have two people participating from Colombia, Monica Betancourt, who is working for Agrosavia, and then Miguel Dita, that most of you might know, who is from SEAT Biodiversity Alliance, but is also based in Colombia at the moment. And then we do have two people from China as well, from the Guangdong Academy of Agricultural Science, Dr. Li Chinyu and Yi Ganyun, and then the last author is my colleague, Dr. Diane Mostert. Right, the content and the discussion today will include a short overview of TR4 and its global dissemination or distribution, the vulnerability of the Ecuadorian banana industry, and then some guidelines how to prepare for TR4, which will include regulation, awareness, biosecurity, detection, and research. I will then also give 4 country examples of TR4 in those countries and how it's spread there and how it's managed over there. And finally, I will conclude with national challenges with TR4. So if we start with the slide right here, something that many of you might know, it was published by Miguel Dita about 2 years ago, is the spores that is probably the most important aspect of the disease pathology of FOC TR4. So what you can see over here are the chlamydospores, which are the survival spores that will be deposited into the soil where they can survive for several decades. And these do infect banana plants, but they can also infect alternative plants and survive on the exudates released by these plants. A second point that I would like to make from this illustration is the fact that there are many other Fusarium Oxysporum strains inside the soils, which can also infect banana roots, but do not cause disease to the banana plant. And we usually refer to these as non-pathogenic Fusarium Oxysporum endophytes. So they remain inside the roots and never progress to cause disease. After the infection has occurred by FOC or Fusarium Oxysporum cubensi, it moves into the rhizome, as you can see illustrated here by the necrotic area through the root into the rhizome over here, that they will colonize before it progresses into the pseudostem. And by this stage, you will see the yellowing that occurs on the leaves, which is different from the yellowing caused by Moko disease, which would affect the youngest leaves. In this case, it will be the older leaves being affected and buckling down, and eventually the entire plant will die. And in this stage, billions of spores are deposited back into the soil. And this is an important factor to remember, that a few spores might initially infect the banana plants, but billions of spores are being deposited back into the soil, which result in the disease becoming severe over the next 2-3 years tremendously. Just a very brief introduction on the different strains affecting banana. We get FOC race 1, race 2, and then race 4. And the fame of FOC race 1 is the damage that it caused on the Gros Michel banana during the 1900s. But FOC TR4 is the one causing the new epidemic that is destroying Cavendish bananas now worldwide. It also affects other bananas affected by races 1 and 2. Right, let's look at the distribution of this fungus. It was first identified when a strain from Taiwan was received and identified as a new strain of race 4 called FOC TR4 or TR4 briefly. But the first damage was caused in Indonesia and Malaysia when the banana export industry in Asia tried to expand their production and moved into Indonesia and Malaysia, and within 6 months, those plantations started to get affected by Fusarium wilt. That fungus was also identified as TR4. It was found in the Northern Territory in Australia over here in 1997. In mainland China, the year later that it was reported, and in Philippines in 2005 when it was reported. So it remains in this Asian area for a very long time, until in 2012 it was found in the Sultanate of Oman, right over here. And this was the first incidence where TR4 actually moved outside of Asia to another country. And after that, there was a rapid expansion of countries affected by TR4. Jordan and Mozambique were next. Jordan here in the Middle East, Mozambique in Africa on the east coast over here. The next year in Lebanon, Pakistan, and importantly, on a second locality in Australia, in the North Queensland, whereafter in 2018 it was found in Vietnam, Myanmar, and Laos. So it actually moved outside of China and into these countries. Last year it was reported from India, a very important, significant development, because India is the biggest producers of banana in the world, and they produce many, many susceptible varieties. So certainly this is a huge challenge for them. And then this year it has been reported the first time from Europe in Turkey, and then from Mayotte, which is a French island on the East African coast. Today, we have the ability to also see where the different strains that we detect in these different countries are coming from, and try to find more or less what their relationship is. So if one look at this diagram or graph, you will see that with the use of sequenced nuclear polymorphic DNAs, one can actually see that all the strains identified near Israel, Jordan, are quite related to each other, while those identified in Myanmar, Pakistan, Vietnam, that are also closely situated from each other, seems to be the same ones as well. It was quite interesting to see that the isolates from Colombia grouped quite closely with the original isolate identified from Indonesia. And I think that will probably be explained a little bit later when stronger technologies will be used. Generally, the introduction of this fungus occurred with planting material coming from an infected country and moved to another country. And once it's introduced, it moves in soil attached to roots or to boots, machinery, and also vehicles working in those fields. And another very important source of dissemination is irrigation water that is being pumped from contaminated rivers onto plantations. So if we look at the Ecuadorian banana industry, and this is a topic that I cannot tell you too much about, but it is very clear to see how important the banana industry is for the country. What is concerning is that a large number of the bananas being produced in Ecuador are actually Cavendish bananas, which means that they are highly susceptible to TR4. The second factor that I do see here is of the 16,000 producers in Ecuador, that many of them are smaller growers. About 61% have less than 31 hectares of bananas, which is something that one has to consider when you prepare for this disease. So I'm just going to discuss a few aspects that you have to think about: regulation, awareness, biosecurity, early and accurate detection, and finally research. About regulation, I think that Ecuador or Agricalidad has done a tremendous job in informing people about the disease, as well as drafting some regulations to deal with the disease once it's introduced, or even to prevent the disease from being introduced into the country. And this includes a national contingency plan, regulation about the importation of banana and plantain, the disinfection at airports and harbors, and the prevention of all other forms of introduction into the country. All of these are available on the Agricalidad website, so you're most welcome to go and look through these regulations. But these are certainly in place for Ecuador. In terms of awareness, I think also that Ursa and several other scientists have done a tremendous job in making the region aware of the threat of TR4. This map over here shows just places where awareness and diagnostic seminars were presented and meetings were held. I know that a lot of trainings had been provided in Ecuador, and even exercises where the possible introduction was simulated, and then trainings to basically show people what to do exactly in such a case. So certainly a lot of work in this regard to make people aware of TR4 in the region. In terms of biosecurity, which is also a very important aspect, and one can see once again Ecuador as one of the 5 largest producers of bananas, shows how important this aspect is. And there are different levels for biosecurity to be implemented. The one is pre-border, before the fungus has been introduced into a country, then activities at the border, and in this case you can see some awareness brochures and boards placed at airports. Over here you can see my colleague Dion Mostert being searched by plant health officials when we arrived in Guayaquil, or in Quito in this case, which is very good. And then post-border off-farm activities, and what I will discuss just now is what the growers themselves have to do to try and protect their properties. There's also a huge difference in the way that commercial growers and small growers will implement biosecurity. And this goes about the material that they use, whether it's tissue culture or suckers. It is important to get that clean planting material for production. The management of people and animals on those farms, access to the properties, the type of production system that you're working with, whether it's a monoculture system, which is highly susceptible, and a mixed cropping system, which might have a little bit more resilience. If you are able to afford and implement sanitation and disease control on your farms, and then obviously the financial capacity of the owners to deal with the protection. Here I just want to illustrate a number of things that one can use to protect your own farm. Firstly, as I mentioned earlier, is to obtain clean planting material. And if this can be coming from tissue culture bananas, this is by far the most important. And I want to stress here that tissue culture bananas, when in vitro, are free of TR4. One does not have to be worried about that. If you have to use planting material suckers, then it is important that it comes from a reputable source and that it is free of TR4. The source of irrigation is very important, because even if you plant healthy plants in your plantation and you introduce the pathogen through your irrigation system, then you can still have a huge amount of damage on your farm. So one has to be sure that the source of irrigation is also clean. And if you share the irrigation system with other growers, that one implements some measures to try and clean up your irrigation water. The use of visible and clearly understandable signs at farm entries, very, very important. And if possible, if you can fence in those farms, once again, for the small growers, this might be a very challenging thing to do. When you clean the vehicles coming onto your farm, it is important to understand also that disinfectants do not work if there's a lot of mud or dirt stuck onto your vehicle or onto the vehicles moving onto that farm. So before you can disinfect your vehicles, it is important to clean it by water until there are no mud stuck to those vehicles. It is important also to manage the visitors coming onto your farm. The best way to do this is not to allow people to drive with their own vehicles onto the farm. This is just a picture at Munapu River Farm, the original Matanushka farm in northern Mozambique, where there is parking available for all visiting vehicles, and people then have to get the boots from the owners and will be transported by the owners onto the property. And finally, the sharing of machinery, equipment, and field tools can also transmit the disease, so it's important to make sure that the equipment and the machinery that you use on the farm is actually clean when it comes onto your property. I showed this picture last year in my presentation in Ecuador, and I'm going to show it again here because this is one of the most crucial factors when TR4 is coming into a country. It has to be reported. If this is not reported, it cannot be hidden. These are just a series of pictures what happened in Mozambique when it was first introduced. This is 1 year after the introduction. That site grew much bigger 1 year later because of the spread of the fungus. And a year later, it was serious damage that was caused on that property. At this stage, it is too late to do anything to try and protect your property. So the quicker you can report that possible introduction and that scientists can come and investigate, the bigger the chances that you can actually contain the pathogen. For Australia, where there are the strictest biosecurity measures in the world, there is a huge penalty if you do not report potential diseased plants in the country. So you have to report it within 1 day for scientists to come in to make sure that your suspect plant is affected by TR4 or not affected by TR4, so that you can actually get the opportunity to try and contain it. If you don't do that, they do penalize growers quite severely. Another point that is really important is accurate identification of FOC. For Ecuador, it is quite important to know that rapid identification usually is done by using molecular markers. But if you get a positive response, it is not good enough to just use that molecular marker for identification. It is important to use a second technology to also try and identify the disease. So it can be VCGs, which is a slightly slower process. It can be sequencing or other types of tests. But a single molecular marker can fail and can give you positive results when the disease is not there. And this was illustrated very well by Freddie McDama in a paper last year, where he actually compared a number of molecular techniques to use for FOC TR4 detection and showed that sometimes your endophytes over here can be detected and can be falsely detected when it in fact is not that specific organism. So it is really important that you have reliable identification of the pathogen. I'm now going to move over to a country where the disease was introduced and not reported immediately to see what is happening in such a country. The country is China, where the first detection occurred in 1998, but about 7 years or 4 years before that, researchers had been seeing plants very, very similar to Fusarium wilt in that country, and nothing was done about that. So it spread really rapidly. In 1998, the disease was found on 14 hectares. By 2010, which is 12 years later, it was found on 40,000 hectares of plants. I visited Guangxi province, one of the major banana producing provinces in China, 2 years ago. They had about 120,000 hectares of bananas planted there. And within 5 years, it was reduced to 66,000 hectares of banana. What is happening in such a case is that people have to find a resistant banana plant, or they have to replace their bananas with alternative crops. In this case, you can see that they replace it with citrus, or they can replace it with corn over there. But for the smaller grower, this is not an option. He simply cannot survive in the plant because the corn does not give him the income that you will get from banana in such a case. I've asked the Chinese people, what are the reasons that it spreads so rapidly and so vastly in China? And these are the things that they mentioned: the large-scale monoculture production of Cavendish in the country, in the banana producing areas. The large number of small-scale farming systems in China. Most farmers are much smaller than the Ecuadorian farms. They're just about 2 or 3 hectares in size. The use of suckers instead of tissue culture banana for planting material. The contamination of the irrigation system and the use of farming equipment that's contaminated from one farm to another farm. Poor quarantine regulations, so people not respecting a national quarantine regulation that's in place. And finally, the poor use of disinfectants at farm gate entrances. So currently, a lot of work is being done in China. I would say that most of the work being published globally on TR4 is coming from China. And one of the centers that's probably best staffed to do this is the Guangdong Academy of Agricultural Sciences. At this institute, they are doing a lot of work on resistant planting material. So they concentrate on conventional breeding, on mutation breeding, and on molecular breeding. And they have the technologies also to do gene editing using the CRISPR-Cas mediated system to do their editing. But they also look at other options: chemical control, biological control, biofumigation of areas, and market development. So a lot of work being done to try and deal with TR from the country. Mozambique is slightly in a similar position. The disease was first detected here in Matanuska. That's a single commercial farm of about 1,500 hectares in northern Mozambique. And then a year later, it was found at Jacaranda farm, which is a much smaller farm of about 50 hectares. These two farms were the only Cavendish monoculture farms in the entire northern area of Mozambique. So the opportunity for spread with rivers or through other susceptible banana varieties was much less. At the moment, these farms are still heavily contaminated with TR4, but it has not moved beyond the borders of Mozambique for the moment or south to Mozambique, simply because of the sparse planting of Cavendish bananas in the region. This illustration is to show how over time the fungus will move. So this is the area where TR4 was first detected at the Jacaranda farm, and it moved over to this plot. As you can see, this is exactly the same group of fungi that has moved first on that farm. And you can see how the vehicles actually moved through wet areas from the contaminated fields around that farm to introduce the fungus into other areas. These spots that you see over here is how the disease developed over time due to the movement of people on that farm. And most of this movement happened before the disease showed any symptoms on the plants. So once it's introduced into the soil, it does not mean that the symptoms will appear within the next month or 2 months. It might only appear after 6 months or 1 year. This graph shows the disease development at Matanushka, and one can clearly see that in the first time, a few months after it was introduced, the incidence was quite low and then rapidly rose quite quickly afterwards. So where it was detected the first time in 2013, by 2015 or 2016, it has increased to about 500,000 plants. And in 2018, this plant was bankrupt due to the severity of the disease. So within 5 years, the entire plantation of 1,500 hectares was destroyed. What we realized afterwards is that the fungus was not introduced in 2013. It was in fact introduced in 2010, most likely. So for 3 years, this fungus had been spreading without it being reported or investigated, and this farm could not be saved in the end. We managed to do a lot of research on this farm before it was sold off on a bankruptcy, due to bankruptcy. And one of the things was testing these GCTCV varieties that were produced in Taiwan. One of these are the GCTCV 218, which was found to be quite partially resistant. And this was then recommended for planting in other fields in Mozambique afterwards. This picture is shown of the Matanushka farm soon after it was sold off on a bankruptcy thing. Absolutely no bananas due to TR4. This picture was taken earlier this year with the replanting of the same farm with Formosana, and this replanting will continue this year. They will probably plant more than 1,000 hectares of Formosana in this specific site. In Australia, you will also find quite an interesting history of the disease. When it was first introduced in Darwin in 1997, it was an area of small size compared to the total production of bananas in Australia. But when it was introduced in northern Queensland in 2015, it was a big wake-up call for them. You could see over here that 95% of all bananas in Australia are being produced in the northern Queensland area. It's very intensive. So all the opportunities there for the disease to spread very rapidly. So just to summarize, for the Northern Territory, detected in 1997, within 6 years, every single banana farm over there was infected, and the disease was declared endemic to the area. The reasons for that fast spread, once again, late detection, the movement of animals, and they have a lot of feral pigs or wild pigs in the area that's moving around, flooding, and flooding is a major reason for the spread of this fungus on a property. It also happened in Mozambique. And this is the situation also with China and throughout Asia, that flooding is moving the fungus really rapidly onto fields. And then the sharing of equipment and planting material. So before the detection in Queensland, and this is the interesting part, legislation was in place. It restricted the movement of machinery and plants from the Northern Territory. So there is no chance that something was allowed to come from the Northern Territory to Queensland. Yet, the growers had little to no farm biosecurity implemented on their farms. They shared a lot of machinery, and they had a lot of service providers visiting the one farm to the other farm to the other farm. These are all very dangerous situations if you do not prepare properly for TR4. Once it was detected in 2015, within 6 months, every single farm in northern Queensland was fenced in. The growers responded really quickly, but too late. So these are just the steps, and I will briefly go through the situation. When it was first detected on a single farm, immediately 10 hectares was killed in an effort to try and prevent the movement of it any further. But in the next 2 years, there were about 8 further detections, And these were random. So you can see over here, this is the farm, and it was just in so many different places that these new detections occurred, that the farm was sold off to the local industry, and they then fenced it in and made it not available for people to visit afterwards. So this entire farm is now not in production for any further bananas, simply to keep the inoculum low. There was also a huge effort to have awareness campaigns and training of growers throughout the entire area. The introduction of biosecurity measures, which also include the control of people and vehicles onto those farms. So everything changed afterwards. They also decided on short-term needs, medium-term needs, and longer-term needs. The short-term needs was the awareness, the extension activities that happened over there, the screening of sanitizers, and this is really, really important, to find a sanitizer that will work effectively and know how to apply the sanitizer and not apply it wrongly. They also looked at alternative hosts that can probably allow this fungus to survive. It will not multiply in this, so it's not a host that will get sick, but it will keep the fungus alive in the soils. The medium-term needs, they identified as looking at the soil to try and ensure healthy soils, to look at the epidemiology and means of spread of the fungus, and finally to have early detection systems and surveillance systems that they try to introduce. And the longer term needs goes back to the screening of material that they can actually plant throughout Australia or northern Queensland if this fungus spread much wider. And the two approaches will be to screen existing planting materials, so existing cultivars, soma clones, and make sure that these are resistant. And secondly, to do some development of resistant varieties. And they use both Cavendish and non-Cavendish varieties and improve them with mutagenesis and screen them to find if they can seize or find something that is of value to the Australian banana industry. The Colombian industry, I think people now know about the outbreak in La Huera in July 2019. Just very quickly, the events that were taking place after the detection. So samples were collected, identified by local scientists. Farm quarantine was introduced. It was also identified by a group outside to make sure what the identity of the fungus was. And currently all banana farms are under surveillance. A lot of biosecurity measures have been implemented and a lot of research is being done, especially on disinfectant in Colombia. So here is also just a summary. One can once again see the importance of diagnostics, of soil health management, and the evaluation of varieties. This is something that can be started in Ecuador right away. So just finally, the difficulty to control the disease or the national challenges is that this disease is not easy to control on Cavendish bananas. It spreads easily, and without people knowing that, you discover the disease only once you see symptomatic plants in your fields. Very often there's a lack of preparedness. I think in the case of Ecuador, a lot has been done to prepare people already, awareness, policies that's in place. But then we have to get the commitment of the stakeholders, of the growers, to buy into this. And this should be a long-term commitment. Lack of funding is a huge problem. One has to have resources to actually fund a response to deal with a disease like that. And then research, which is really, really important to do even before the fungus is introduced. I'm going to end my presentation with this slide being provided to me by Biosecurity Queensland, where they indicate that in 2015, TR4 was first detected in a very intense production area with a lot of bananas in North Queensland. Until now, only 5 farms were affected by the disease. So they were able to contain the fungus tremendously well. And that was due to a number of things: good regulation, industry commitment, and research. Those 3 pillars helped them to deal with the disease in the country. So it's summarized over here: the success is largely due to growers, government, industry, scientists, and researchers working together. And with that, I thank you for listening. I'm happy to answer some questions.
Agradecemos a Altus Viljoen por su intervención. Tenemos algunas preguntas de los asistentes. Vamos a dar lectura a las mismas. A ver. ¿Qué desinfectantes han encontrado como más efectivos para la prevención y control del TR4?
So the most important ones are the quaternary and there is one product that has been published very often in the literature by my research group, as well as the researchers in Australia and other researchers, which is, I'm not sure if I can say the name of here, but it's in the literature, scientific literature, so I think it's public knowledge. A product called Spore Kill, which is probably the best product that you can use. I think also what is important for me to mention at this stage is that these disinfectants do not work if there's a lot of soil attached to your vehicles or to your shoes. So they have to be cleaned first before you can apply the disinfectants.
Bien, siguiente pregunta. El sector bananero de varios países de América Latina ven la necesidad urgente de ingresar materiales resistentes o tolerantes al foque R4T. ¿Cuál sería su recomendación?
Right now, I think the Cavendish material that's available is limited. So there are some summer clones that were developed in Taiwan. I also know that there's quite a bit of mutation breeding happening globally. And I'm aware of the work that's been done by Rahan Meristem. But what is right now being used by countries that is dealing with TR4 in their fields and need to replace their bananas is the Formosana. That is now being planted widely in the Philippines. It's being planted widely also in Mozambique. And I think it's being tested in many other countries at the moment. I hope that within the next year or 2 years, more options will become available. There is a lot of work being done in plant improvement globally at the moment. There's a lot of work being done in mutation breeding in China. So I think that hopefully within a year or 2 years, there will be a choice for growers to plant some, let's say, partially resistant material or tolerant material. And I think if these become available, the moment that they are available, they should be tested in your country. I think one of the disadvantages of somoclonal variants is that they do perform slightly different in other countries than in the country where they were developed. So it is important for Ecuador to test this material exactly the same way that Colombia is now going to test it.
¿Cómo pudo haber llegado el virus de Indonesia a Colombia? ¿Cuál es la teoría mejor adaptada a la realidad?
I have absolutely no idea. This is work that's been done by a group in Israel. I do believe that we need stronger techniques to confirm this. This has been done with a limited number of strains that they included in their study. And it's been tested with a single technique. But these days with whole genome sequencing, the technologies become stronger and stronger. And I think that in a year or so, we will probably see if this is indeed the case. More strains probably also have to be included from both Indonesia and Colombia to see what the situation is. I think it would be unwise for us at this stage to try and suggest that the fungus was introduced from Indonesia into Colombia only based on this work.
Dejando a un lado el Cavendish y tomando en consideración el banano orgánico, al trabajar con micronutrientes, bacterias, cobertura vegetal, ¿resulta la propagación del R4T aún muy agresiva? ¿En qué tiempo se estima que salga un producto que pueda combatir al R4T?
I'm very optimistic about maybe things like bionutrients that you can put into the soils, and specifically about ground covers. And I do not know what is the best ground cover to use, but the reason why I am positive about ground covers in a banana plantation is because it does stimulate other microbes that can compete with your pathogens in the soils. But more importantly, it prevents the movement of soil in that specific plantation with people walking over that soil, maybe erosion due to water running on that soil. And as I mentioned a little bit earlier, when the fungus is introduced, you don't know that. You only see it once the symptoms start to develop. And by that time, there can be a large spread of the fungus already in your plantation. If you have a ground cover, it will reduce that spread in your plantation tremendously. So any ground covers being used in plantations, I would absolutely support.
En las fincas bananeras, donde ya se encuentra el hongo del Fusarium, ¿qué otros cultivos alternativos podemos utilizar en este suelo ya contaminado?
Alternative crops to use in Ecuador? I guess you have to ask some people in Ecuador. Certainly there are some other options. If it's going to be as financially rewarding as bananas, that's a different question. And that makes it difficult for small growers in such a case. But I'm sure there are other crops that they can plant. We do not know at this stage what is the status of plantains against TR4. So certainly this is one of the first type of tests that need to be done.
¿Cuáles son las dosis recomendadas actualmente y qué producto utilizar para las desinfecciones? En muchas de las fincas en el Ecuador se sigue utilizando el hipoclorito de sodio.
Yes, I would not use sodium hypochlorite. There's a lot of environmentally damaging reasons not to use sodium hypochlorite. As I said, and I guess I can mention it simply because it's in the literature, I will use the product Sporkill. It is a tertiary ammonium compound. There are publications by the Australians. I think it appeared last year in Plant Disease, where they have compared a large number of disinfectants. I know the same work is being done in Colombia and several other countries. And every time your tertiary ammonium compounds will be the ones that work best. But of those, I know that sporkyl is being used widely in Australia. It's being used widely in Southern Africa at the moment. And it's working very well. And if you use it in your water baths or even your food baths, they do retain their activity for quite a while. In Australia, they indicated quite long. I would not say one should use it for a week or more, but in South Africa, we use it for at least 3 days before replacing the water. The big thing is it cannot penetrate, and it's not going to work when something is very muddy. So, and none of the other products will work for that either. So you need clean surfaces to actually disinfect material against TR4.
¿Qué resultado se ha obtenido acerca de la investigación a los suelos supresivos? ¿Se debe evaluar materiales nativos para el control de Fusarium?
Yeah, we've done quite a bit of research on suppressive soils, and quite a bit of work has been done around the world. At one stage, the organism most commonly associated with suppressive soils were the non-pathogenic Fusarium Oxysporum, which is an interesting thing, but it makes also quite a bit of sense because that also stimulates the response of the plant to try and protect itself eventually. There were some other organisms that also worked, especially the bacteria, that were not bad at all, and the actinomycetes. The problem is, in the end, how to reintroduce these organisms and get them to compete with the pathogens in the field. And the best way of introduction would probably to establish these as endophytes in the plant, but then they have to survive over a long time, and they have to colonize that plant very well. So there's a lot of challenging with biological control. There's also a lot of work being done at the moment with communities of microbes in the soil through microbiome studies. And I think this will probably result in some products that can eventually help. I know that the people in India is doing a lot of work. They strongly believe in trichoderma and the introduction of trichoderma into plantations, as well as some bacteria. So I do not think this is something that one should not consider when trying to manage the disease. But that this is an absolute cure for the disease or a complete solution as a standalone, I'm not sure if this is going to work. You will have to bring in tolerant plants or partially resistant plants combined with these biological control products in an integrated disease management approach.
A partir de los avances tecnológicos, los investigadores han desarrollado sistemas de inteligencia artificial que pueden tomar o captar fotografías que determinan la enfermedad que tiene la planta. Esto, aparte, arroja ciertas recomendaciones. ¿Qué opina usted sobre estas tecnologías o estos avances tecnológicos para apoyar al proceso de los agricultores?
Yeah, I'm very happy about these developments. I do think one should not make conclusions too quickly because there are a number of stresses and even abiotic stresses that can cause symptoms or plant responses similar to banana Fusarium wilt. Typical thing is the leaf yellowing that one can be caused by other organisms as well. So that there's no confusion in the way that these intelligence systems actually detect the response of the plant to try and monitor there. But I think this is a developing science, and hopefully soon, within the next year or 2 years, there will be breakthroughs that can help tremendously in the fight against Fusarium wilt in general. I know that the Australians are also doing this, and I think many other people looking at volatiles and the use of dogs to actually smell plants that are infected with DR4 to identify that long before the symptoms appear. And I think this is an exciting area as well. It's an expensive type of experiment, and it's difficult as well, because you cannot test that afterwards, for instance, like COVID, that you can do a test afterwards to find out that someone is indeed infected, even though that person does not show the symptoms. Plants do not talk, and it's quite hard to try and get that control test there. But this is work that's being done, and certainly all ways to do early identification of contaminated plants, that's the right way to go.
Si no hay más preguntas, les recordamos que pueden compartirlas con nosotros a través del chat y se la transmitiremos al profesor Altus a efectos de que pueda darles una respuesta en breve. Bien, mi estimado Altus, muchas gracias por su asistencia y a todos aquellos que han participado de la conferencia.
Thank you very much for the opportunity, and good luck with the rest of the conference.